I'm going to focus my talk today on pancreatic and breast cancer, really, and the target that I'm going to talk about, some people may know, some people may not, is a mucin. It's called mucin 1. And I'm going to then talk about the development of a novel antibody, preclinical data on targeted therapy, and clinical data on the use of this antibody to detect circulating tumor MUC1 as early detection. So significance, obviously globally, you've been hearing this all the time, there would be 1.6 million women will be diagnosed with breast cancer this year itself, and many will die, succumb from this disease. And 20% of all of these invasive breast cancers are triple-negative breast cancer. That means they don't have any hormonal receptors, and therefore can't be treated by the hormones. And it's a number 2 killer amongst all cancers in women in the US and I think even in the world. Similarly, there are many people who die of pancreatic cancer. Pancreatic cancer is basically a lethal disease. And it's a number 4 amongst all cancer deaths. So what's the commonality? The commonality is both of them arise from the ducts of your glands. So they are epithelial in nature. They arise from the ducts of these 2 organs. So that's one commonality. The other commonality is that these organs actually express a protein called MUC1. And this protein in a normal epithelia will always be on the apical surface, will have a lot of sugars attached to it, and therefore the core protein will be hidden. However, what happens in adenocarcinoma is it changes. It gets overexpressed and it gets less glycosylated, so it becomes foreign to the body. What is MUC1? MUC1 is basically a glycoprotein. It stands like a bottle brush on the cell surface. It has a cytoplasmic tail, which is extremely potent in oncogenesis. But what I'm going to focus on really is the top part of MUC1. And I'm going to go a little fast because of time. But as I said, post-translational modification of this MUC1 makes this MUC1 very aberrant. So when it becomes aberrant, it becomes a target as well. And that's what we're going to focus on. So what are the clinical applications? Are there clinical applications? So in Science magazine in 2012, it was dedicated to mucins on epithelial cells and how many diseases it is associated with. So MUC1 was ranked by NCI as the second most best target out of 75 tumor-associated antigens, and this was done by NCI for the development of a vaccine against this tumor form of MUC1. And there are 30 clinical trials ongoing on this particular molecule. One of the vaccines which actually we tested in the lab is called Stimuvax, and Merck took it over, and now it's in phase 3 clinical trial, and for small cell lung carcinoma. So what needs to be done really? How do we study this molecule? You can't always study it in a human being. So what Dr. Sandra Gender did was create a mouse model that actually has human MUC1 in it. It doesn't have the mouse MUC1, it has human MUC1, and the expression, remember it's a transgenic mouse, but the expression is normal, so it doesn't have overexpression. It's not a model of overexpression, it's immune competent, and if now you mate these mice to any of the oncogenic mice, what you get is a mouse that develops a tumor in the organ. That means the hormones, the stroma is all correct, and the MUC1 changes as the tumor develops. So the normal MUC1 becomes aberrant MUC1. Now you have a system where you can really use this model very well to do all kinds of things. The immunological studies, targeted therapies, and study tumor progression without going into the human. Of course, it's not a perfect model, but it's a great model to study many things. So this model is a model of pancreatic ductal adenocarcinoma. So you have the KRAS mutation in this mouse. It develops pancreatic adenocarcinoma. It has p53 suppressed. It has MUC1 altered. It has appropriate stroma and hormone, and it's immune competent. This mouse, when mated to MUC1 transgenic mouse, will develop these Pannin lesions. Just like humans, it will develop Pannin lesions and then adenocarcinoma. So what we were able to do was develop an antibody using this particular tumor. So the tumor-associated MUC1, at the stage when it was changing from normal to tumor, that's when we used the lysate from that tumor and made an antibody. And I didn't know what I was going to get into. The reason I was making an antibody was to use this antibody to target drugs to the tumor. Because systemic chemotherapy is pretty bad, systemic immune therapy is pretty bad, every therapy that is systemic is pretty bad to the body. You can't give enough of the drug, and that's one of the problems. So that's the reason for development of this antibody. So this antibody recognizes an aberrant form of MUC1, which I told you, the tumor-associated form of MUC1. It's very unique, and what needs to happen, it has to have that silyl TN attached to a core protein. Otherwise, this antibody doesn't recognize the antigen. And 95% of breast cancer expresses this antigen, and 80% of pancreatic ductal adenocarcinoma expresses this antigen. So it's huge. Actually, the target is huge, and that's why it's the second best target, as NCI puts it. So here's an immunohistochemistry with that antibody, and you can see that it doesn't stain the normal breast, and right from pre-invasive DCIS, you can see really nice staining. And you can also see that in this tissue, even the normal tissue right beside it doesn't stain. Only the cancer tissue stains. This is again exemplified by using benign breast, the fibroadenomas, inflammation, all of that is negative. But if you go to DCIS or invasive breast cancer, you get this nice brown hued staining. And the oncologist who was coding this, we've done a lot of them, 368, and actually done by Lokamudra Das Roy over here. And Chad Levesey, who actually looked at it, said, oh my gosh, I can just pick up this and not even have to look under the microscope to say if it is benign or malignant. He was that impressed with it. So similarly, with, in pancreatic cancer, we saw the same kind of issue. We saw really strong and very precise staining just in the ductal adenocarcinomas. So now I'll move on to say that, okay, it's present, it's wonderful that it's present on the actual primary tissue, but is it also present in the cancer-initiating cells, which is the cancer stem cell? And lo and behold, at that time, there was an article written by somebody else that MUC1 is actually expressed on pluripotent stem cells. And NCI actually said that MUC1 is expressed on some of the cancer stem cells too. So we wanted to see how good this would be. And so what we did was we looked at several pancreatic cancer patients, dissected fresh just when the Whipple was happening. We took the pancreas, actually not me, my postdoc at that time, Dr. Jenny Curry, and she actually digested it and at that time looked for these pancreatic cancer stem cells and said, does our antibody recognize it? And lo and behold, it does. Almost all of the patient cancer stem cells was stained with this antibody, which was really great news for us so that we can use this now. This is the demographics of it. So now the question was, can we target these pancreatic ductal adenocarcinoma cells and the stem cells with something? Can we leverage this antibody to do this targeted therapy? So the way my graduate student decided to do it, he said, instead of conjugating just the antibody to a drug and driving it, he said, why don't I use a nanoparticle that can put a whole load of payload of chemotherapy in there, conjugate that to the antibody, and then drive it to the tumor. I said, do whatever, do both ways and see what works really. Because if you just use the antibody, you can imagine you can only put a little bit of the drug on that antibody. But if you have this nanoparticle, you can just load up a huge amount of chemotherapy in there, and you can put different chemotherapies. So you can use multiple chemotherapies in there immunotherapies, all kinds of things in that nanoparticle. Plus, it's safer to administer. So here's what he did. He kind of used a nanoparticle, and this is not to scale by any chance, has a linker which is proprietary, and then added the antibody to it. Now what he did was he used some of the cells and looked for its internalization or its surface staining. These are just some pretty pictures showing that within 30 minutes or actually 60 minutes, he actually sees the antibody going into the cell. And this is best exemplified in this picture where you can see that very beautifully the antibody internalizes within 60 minutes of staining, of putting this particle into tissue culture. And these are just cells that do not have the MUC1. So they are negative for MUC1, so you can see how specific it is. It doesn't stain at all. Here is again where he did another cell line, and he looked at that, and you can actually see what he did was he has compared nanoparticles alone or nanoparticles that were conjugated. And you can see the difference of how beautifully this is internalized and this isn't as much. Although nanoparticles themselves will go in, but not as much if you have a real target with the nanoparticle. Again, this is showing it on a different cell line. Again, you see this beautiful image of this. So, of course, the next step was what? Okay, throw it in and see if it is better in killing the cells in vitro. So that's what he did. And I'm sorry that these are not all, it's not always a matter of how much you put in. We are learning very much that this MTS assay is not the best assay in the world. And also, just adding more drug or more nanoparticle doesn't mean you're going to get better results. So the best results we actually got was at the lower dose, where you can see that this is nanoparticle alone with the drug. It's paclitaxel in this case, I think. Yes. And the green is nanoparticle with paclitaxel and the antibody. So you see, there is a big reduction in there is more depth of these cells when you have a target. That means it's internalizing more and it's killing the cells more. So this was pretty promising. And for another cell line, BXPC3, which is very, very sensitive, we really didn't see any difference because it's already very sensitive to this nanoparticle. So we don't see it. It works better if you have a chemo-resistant cell line, which is, again, a good thing for us. And this he was using whether, there's a gamma secretase inhibitor that's used for targeting cancer stem cells. So he used that. The data isn't all that great, but you can see that if you don't have MUC1, you don't see any killing, but if you have MUC1, you see some killing at least of the cell lines. Of course, the proof is in whether it's going to work in vivo. So now using these mouse models that I described, actually what he's done is he's actually used this mouse model and created cell lines. And now he orthotopically injects them into the pancreas. He creates an orthotopic injection, and he has luciferase in that cell line. Now that luciferase will look like this when you image it on an IVIS system. So here's your tumor, and here's your antibody that is conjugated to just a fluorochrome. It's called isocyanide something, green. Anyway, ICG is just a fluorochrome. It's an FDA-approved fluorochrome that's used actually in clinic for some other reasons. So you can see that in 24 hours, almost all of that is in the tumor. And don't forget, these mice actually are MUC1 transgenic mice. So the entire body, all of the epithelia actually has normal MUC1. Now here is a nanoparticle with ICG. Here's the same tumor, but you don't see that nanoparticle localizing on the tumor. So now next one, which was the most interesting one for me, is here's the tumor, here's the nanoparticle with the antibody. So you get a beautiful, just exactly where the tumor is, you get the localization of that. So now we know that we can actually take this formulation, put it in a mouse, and it will not go anywhere, hopefully not go anywhere, just go to the tumor. Here's just, he took some organs out, and you can see that, the tumor just shines up like that. There is some activity in the liver, of course. That's where everything goes when you do an injection. And here's the nanoparticle with ICG, which did not have that TAB004 or that antibody attached to it. How much time do I have? 2 minutes. Here again, I'm showing you the same thing, pancreas. You can even do that with breast. This is a breast tumor model. And when you open the mouse up, you can actually just see it in the pancreas. It's just really, really nicely located. I'm going to go a little fast, and I'm going to tell you what we are doing now with, so that was more therapeutic. Now what I really want to get at quickly in 2 minutes is that we can act this MUC1, when it's in the tumor form, actually sheds into circulation. So it is used as a biomarker in the serum. It's already used, CA15-3 and all that kind of stuff is used. But because this antibody turned out to be so specific, we thought, let us try it and see what happens. Now, when this sheds into the tumor, we have a simple ELISA, and what happens is all of the normal ones goes away, and only the tumor form actually attaches to the antibody, and we get a really nice result. And again, Dr. Lopamudra Dasroy did a huge randomized trial with many patients, and what she saw in the initial study was a very nice differentiation between malignants and benigns and normals. So here are all the benigns and normals, and here are all the malignants. These pink ones are actually DCIS. Of course, there are some that are false positives in this case. Again, what I want to stress at is that this antigen actually goes up as the stage goes up, which is very important to know. And what Oncotab has done, the company has done, is actually licensed this technology from UNC Charlotte and made it into a actual test for women to use, or for physicians to use, primary care physicians, mammogram centers to use in patients, because there are women, we've heard a lot about breast cancer and mammograms. Women who have highly dense breasts, 50% of them, mammograms do not work. They just don't work. It's like looking at a snowflake in a blizzard, because it's white on white. So it doesn't work. And whatever anybody says, it doesn't work, and they get diagnosed only at stage 3C, after they detect a lump by self-examination. So for them, self-examination is much better than mammogram. So basically what we are saying is, using this Alcura personalized score, if it is positive, if it's tending high, then you would go and do every 6 months. If tending not high, you do every 6 months this test, or with your annual mammogram. If it is yes, then you go and do additional imaging and biopsy. Again, in pancreas cancer also, you see that by stage it goes up. And we've already published that. So in conclusion, what I've shared with you today very quickly is that TAB004 detects bound MUC1 on cancer tissue, but not on normal tissue. It detects shed MUC1 in the serum of mice and patients with breast and pancreatic cancer, and it's stage specific. So the relevance is early detection of these cancers before the cancers become, and especially for pancreatic cancer, we detect it at the stage of panning 2A lesions. So it is very important to note that we can probably have this test available for pancreatic cancer as well. So again, to reiterate, TAB004 detects MUC1 on cancer stem cells, in vivo xenografts, and in patients with pancreatic cancer. TAB004 can be used with drug-loaded nanoparticles in vitro and in vivo. So the relevance, again, it's the second most targetable antigen by NCI standards, and it might be possible to target cancer stem cells, which are really the problem, because once you remove all the cancer, it's the cancer stem cells. Okay, I'm done with my time. And here's the path through biotech. If there are young people who like to discover, this is what you should do. Immediately patent your technology. We have 2 patents already issued on our antibody and the uses for it. And then you build your company and do whatever you have to do. So this is what we are doing: blood-based diagnostics, in vivo imaging, combination targeted therapeutics. And in the therapeutics, we have 3 things we are doing: TAB-004 combination, TAB drug conjugate, and TAB CAR T cells, which I'm not going to talk about. And that's the end of my presentation. Thank you very much.
Thank you for a nice presentation. Any questions for Dr. Pinku Mukherjee?
Yeah.
Very interesting and a lot of promise. But my only concern is that pancreatic cancer, they're well known for their fibrotic nature.
Yes.
They're very fibrotic. And that's one of the reasons they feel that the drug is not able to penetrate into the tumor cell.
Yes.
So how do you explain that these nanoparticles are capable of getting inside the tumor cells? Is your tumors in this model palpable?
Yes. It's palpable tumors. As you saw the tumor inside, and the orthotopic ones are really palpable. I showed it in 2 models actually. In the orthotopic, where you actually create it, and the other one is actually one that develops spontaneously.
So how do you see the future in patients? I mean, those are fibrotic tumors.
Yes, but it goes there. I mean, it's not like it doesn't go at all. And I think what happens is, because this antibody kind of directs it right there, and then, maybe gets internalized over there. I don't know exactly how that happens, but it happens. And the main thing, I think, the way we want to do this is actually give it a chemotherapy before, so that it becomes leaky, at a suboptimal dose. We've done that. We've done actually gemcitabine at a low dose, which doesn't actually curable optimized dose, and then we put this in. So, it works better that way.
And what are the inducers of this MUC1?
Inducers? Yeah. There are many inducers. It's actually the pro-inflammatory atmosphere, and you probably know more about what induces the MUC1. But there are many things that induce MUC1 to become this. So MUC1 is always produced. It's a part of your normal epithelium, right? So it's always there. It's produced as a part of your epithelia that for lubrication, for attachment of cell to cell, and for forming the gel that you have in all of your organs. So that's what MUC1 is for. So it's always produced. So it's not like it's being produced only when there is cancer. What happens in cancer is just aberrant. The post-translation modification that occurs because of the glycosylation changes. So the glycosyl transferases, they have some mutations, or they are not active correctly, or that's what the basic thing is, that that glycosylation of MUC1 actually changes. And because that changes, and the cells now become all around, so they lose apical, their structure, and so the MUC1 now comes in contact with a lot of the growth factors as well. So that makes it more oncogenic as well. So it's not the actual process of creating MUC1 that changes. It's the post-translational modification.
Nice presentation. I would like to ask, what are the negative effects of these nanoparticle side effects when we are introducing something new into the tissues?
Yeah, so here's what we have seen. So as you saw, by 72 to 96 hours, we don't see it at all. It's gone. So it's gone. So and these are all mouse experiments, so they haven't put it into the clinic yet, right? So in the mouse, plus these are PLGA nanoparticles, which are the same things that are used for sutures and things like that, so they kind of dissolve by themselves. I'm more worried about what happens to the antibody. If the antibody stays in, what happens? But there is precedence that a lot of antibody therapy is already ongoing.
I had another conference I attended where they had introduced nanoparticles, and there was a massive liver damage. in one of
Massive liver damage?
Yeah.
Okay, so using the, as you saw, using the antibody, it really doesn't go much to the liver. And even if it does, it clears very fast.
No, that was a liver problem, okay. And it induced a huge liver damage, and even photographs were shown.
Mm-hmm. Possible. I don't know what nanoparticles they were using.
That I don't remember at the moment.
But you know, liposomes are already being used, right? I mean, Abraxane is albumin, liposomes are being used. There are many others that are being used in the clinic with a lot of efficacy too. So it depends on if you're using a silica-based nanoparticle that just doesn't clear at all, and we've done that. It doesn't clear for months from the liver. There you are going to see damage. But you know, when I speak to oncologists, they say small amount of liver damage is nothing compared to what they go through in pancreatic cancer or triple-negative breast cancer. I mean, the kind of therapy they are getting, it's damaging not just the liver, but a lot of other things too.
Okay, thank you.